Well, I'm kind of excited not only to end on, polymers and biological macromolecules, which is just another word for polymers, but I'm excited because it's polymers. It's something I enjoy talking about. It's a very interesting area of research going on right now and making materials through not just use of polymers, but maybe polymers and other types of nanoparticles, ceramics, just some really cool stuff going on in the area of nanochemistry, or I guess chemistry at the nanoscale level, where we're trying to engineer molecules to make certain constructs that are larger than just the six carbons. It might be 60 carbons, 600 carbons, 6,000 carbons. Kind of bottom up chemistry, they say, going from atoms to molecules to nanostructures to macrostructures, microstructures, micrometer. Things like the biology does. Biology is a awesome, mother of chemistry. Gives us a lot of leads on what we can do better in chemistry. And so I have on this slide this statement that I don't want to make fun of a chemist. We're doing a great job, but compared to what nature does, just without any guidance or any aid, the constructs that it makes are pretty dang amazing. So if you want to check out a video, there's a link in the PDF form of these notes. It's really good. It talks about just the simple task of protein synthesis from DNA. Just really, really cool stuff. What we're going to do first is just talk about the basics of polymers, terminology that is around polymers. Then talk about the biological polymers or biological macromolecules. And I'm not going to talk about RNA and DNA. That'd be another type of macromolecule. But we're going to talk about the three others. Lipids, which fats, triglycerides, carbohydrates or polysaccharides, and polypeptides or proteins. So it's really like there's four biological macromolecules: RNA, DNA, we have proteins, carbohydrates, and lipids. But we're just going to talk about three of those. Now, just to kind of get you excited about where we're going, so it turns out if you take this protein sort of folded up on itself in secondary, tertiary, quaternary structures, if you've taken biology, there's a residue that's very important in the activity against, a cell, a bacterial cell. Penicillin will take to this particular site in the cell wall of that bacteria. It'll sort of destroy it in this way. This is called a beta-lactam ring. This ring right here is called a beta-lactam ring. And what happens is it's got some strain in it. There's a video that you can watch. I believe it's hyperlinked underneath this figure, but it's strained. This ring, four-membered ring, is not favored. You'd rather have 5 or 6, but more so, you have two sites that are really not, they're not very stable. This carbon here is very, very partially positive, so I'll put that in blue. And you would think, if you've taken a little bit of chemistry, or if you take organic chemistry, you'll know that nitrogens next to a carbonyl often will feed in electrons to kind of pacify or sort of settle down that partial positive charge on this carbon. It'll give electrons, it donates. But this one can't. There's a reason why, because the four-membered ring. So anyway, it makes that carbon very susceptible to partial negative charges nearby, this being a good candidate, this site on the protein, on the cell wall. We can take and have that react, this oxygen start to bond to this carbon, and it blows up the beta-lactam ring, breaks it like a bomb, and it then stops the growth of that bacteria, because now this penicillin drug is bound, to this sarin residue of the pocket, blocking or preventing the continuance of that bacterial growth or whatever. So it's kind of neat stories that you can get into talking about the macromolecules and their pockets within proteins or within cell walls. Just some of this chemistry that we have with biological macromolecules. So here are some terms. Poly means many, many mers, many monomers. So When you hear polymers, just think many of something, repeating along the way. The basic structure is to have, let's say, these little turquoise boxes then would be the mers, and they can be very linear, just like a spaghetti chain, or we can have what's called branched polymers, where we have some spaghetti chain, but off of them are some more monomer units. You get this with like amylopectin, part of your starch has sometimes the amylose is linear, but then you can have another part of the starch, carbohydrates that are branched polymers. You can also have, like with your egg yolks, you'll have sulfide linkages that connect two polymers to each other through a cross-linking. And it still be flexible, like your rubber bands have a little bit of cross-linking between the polymer chains, so it has a little bit of ability to snap back. Have too much cross-linking and it becomes rigid. We can have all the way, kind of, if you want to think of cross-linking between chains, it's so pervasive, you really don't have one chain or another really defined that you can see in a few cross-links. It's all what we call a network solid, kind of like glass or diamond. You can think of it that way. All these linkages, well, sometimes in polymer chemistry say you have like B, B, B linkage. There's three ways it can bond, and let's say it bonds to A sites, A, and A. So if you were to put this together, it would just grow into this mass rock, kind of like material that's very, very rigid. We call that a network bonding. You get this in some of your lignins, in plant life. You get this kind of network solid, where it's not directional, the polymer chain growth or the growth in the macromolecule. But we see the disulfide linkages of proteins. This is that amylopectin. I'm not probably saying it right, but it's part of their starch. You get some of this type of branched polymers. And then the amino acid sequence goes into polypeptides. It would be a form of a linear polymer. There's hyperlinks to each of these. You want to check those out, the stories behind each of these examples. Oh, forgot to mention, this is so cool. So if you're doing polymer chemistry in the lab, you typically, synthetic polymer chemistry, you don't have the ability to take each segment and define it as it's, these turquoise boxes here. They just kind of come together randomly. But proteins get this specificity where every single mer unit is set to a certain identity. And we have 21 amino acids or whatever. So you can actually set that code, you might say, for your proteins. Well, we know that is set by the DNA, transfers it to an RNA. So you get this, again, not just stupid, I'm going to say dumb polymers, but you get polymers that, A, B, C, and then A, just there are different units that come together in a very particular way. That's a very smart material that we have for proteins and for DNA and RNA. Those types of macromolecules are very, very smart, you might say, very, very complex. All right. I'm going to try to go a little bit faster, some links as you go to the PDFs for some of these examples that I pull these figures from. So there's two types of polymeric systems in the body, and then I'm just going to talk about two types of polymeric systems that we have for synthetic labs, or not biological-based polymers. So the first one is just our sugar. We can have a glucose molecule here, and we can have what's called a 1,4 glycosidic linkage, where the oxygen off of carbon 1 will bond to the carbon of another glucose molecule at carbon 4, and we'll get a 1,4 glycosidic linkage with water loss. So this is going to be a condensation polymerization, because we get this plus water lost. And it's kind of a step growth polymerization, meaning it just kind of click, 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 and it keeps growing kind of slowly. In our time, I guess on the time scales of polymerization, it's a little bit slower than what we're going to see on the synthetic side of polystyrene cups and polyethylene bags. It's called a condensation polymerization because we're kicking off water, and it's kind of a step growth polymerization. The linkages, you can have up to like 3,000 repeating units like this within that chain. Another type of step growth polymerization involves amino acids, where you have the amine and you have the acid group, hence the name amino acids, and you have that group off of the amino acid, the R group, that can be very different between all the various amino acids. But we make, if we take the carboxylic acid group of one compound and have another one just like this, but written right here, then we have the amine group reacting with the carboxylic acid group of another monomer, and we get the very important amide linkage, what we call a peptide bond. You'll notice this nitrogen here has lost one hydrogen, and the carbon here, would be this one right here, has lost an OH. It still has this oxygen here. So this would also be a condensation polymerization because we've made this plus water. Both of these then are condensation polymerizations. Both of them are step growth polymerizations. When we go to the synthetic side, meaning like what chemists do in the lab, without biological proteins, this is you need to make sure understand this is in the body. It's probably facilitated by a protein, so it grows in particular ways. We know for amino acids to polypeptides, that's facilitated by RNA, a code. But here we just throw in with a initiator ethylene, and the initiator pops it on or turns it on, initiates, then we get bzz, propagation, and this thing happens really, really fast. Compared to these types of polymerizations, this is not much faster. We call it a chain growth polymerization. You kind of think of it like a zipper, just zips real quick and it shuts off after very, very fast propagation. It goes through a termination. We can make from ethylene, we can make polyethylene. From styrene as our monomer, we can zip it on very quickly to itself through initiation and make polystyrene. Put a lot of air into that and you get a polystyrene cup. So we have a lot of commodity polymers made from just these two types of polymers. Polyethylene, you have everything from your Walmart bags to your milk jugs. We have polystyrene used for your, of course, styrofoam cups. Just very prevalent in the market, these commodity polymers. So we said there's 4 types of biological macromolecules. The amino acids we talked a little bit about, fatty acids, the carbohydrates made from simple sugars, in the case glucose, galactose, fructose, and then we have nucleotides that are used to make DNA and RNA. I just wanted you to kind of remember, we're going to talk about just 3 of those. We're going to talk about just this side, not the DNA and RNA types of macromolecules. So first of all, lipids. Well, we can make, so we have triglycerides, also called lipids, that can undergo a they can undergo a deesterification. This is an ester type of linkage. I want to specify this is an ester. If you have a carbonyl, C double bond O, with an oxygen off of it to another carbon, that's called an ester linkage. Well, we can take a protein, lipase, and we can cut that bond, that ester, so that we cut off this oxygen and we replace it with water. So it's a hydrolyzation, not a condensation, but we're hydrolyzing this so that the oxygens here, kind of think of it as one of those hydrogens here in H2O goes to this oxygen coming off, and the OH left goes to this carbon. So of course it's going to take 3 water molecules to hydrolyze this ester or this triglyceride to the fatty acids. You need to know that these are carboxylic acids. All these here are carboxylic acids, and you can have different R groups here within, depending upon what your triglyceride looks like as it starts. This is actually the protein lipase, and they're proposing that the way this happens in the body, you have a lot of partners getting together to help this reaction go. We've got a carboxylate within the protein pocket, so you can kind of think of this maybe as part of a protein, where these pieces, these residues, have a part in the protein pocket of taking our triglyceride and breaking it down. So we have a histidine unit. It's going to grab the proton as this oxygen is attacking the carbonyl. we can have this oxygen now gaining electrons as this guy is attacked by this oxygen. We have this oxygen taking one of the protons from lutein or from a nearby amine. We can have this oxygen protonated. So it starts the process of hydrolysis of the triglyceride. I haven't finished it here, but it just kind of shows you in the body that there's a lot of players involved within this transformation of this triglyceride to fatty acids. All right, so that was our lipids going to carboxylic acids. We can have two glucose molecules. We talked about that 1,4 glycosidic bond. This is carbon 1. We know it's in the numbering system, always carbon 1 is the one in the 6-membered rings that's bonded to 2 oxygens. So here's one here, and here's one here. So in the numbering system they have for monosaccharides that are 6-membered rings, carbon 1 is always the one that has 2 oxygens off of it. So we can kind of say, well, that'd be over here, be 1 then, because there's an oxygen and there's an oxygen. But this would be then counting from 1 to 2 to 3 to 4. And we're going to lose water here, so there's our condensation, the green. And we're going to have the linkage between this oxygen and this carbon here. And we're going to notice, this is something really important, that the oxygen here in red, in terms of this ring, is on the opposite side of what we have as a CH2OH group here. This is called an alpha linkage. An alpha 1,4 glycosidic linkage. Alpha is in respect to this oxygen that's linking these two sugars relative to that CH2OH group. This is a CH2 group there, and that OH. If it's on the opposite side of this overall ring, then it's an alpha linkage. Well, we have a lot of those here then, so we can just start repeating this sort of alpha 1,4 glycosidic bond over and over and over again. Have about 300 to 600, even up to thousands of these repeating itself, so that we have this CH2OH again on the north side of the ring, and this is on the south side, opposite side. So we have a lot of alpha 1,4 glycosidic bonds being formed. We can just keep repeating that, and we can make not just a disaccharide, but we can make a polysaccharide. Amylose is a type of as a part of our starch carbohydrate. There's amylopectin that is also a part of starch. Anyway, you can also then have what we call maybe sort of like secondary structure. This amylose can start to fold on itself to make helical structures. We're almost done. Just wanted to say you have two types of really polysaccharides. Those that have an alpha 1,4 linkage would be part of your starch, but cellulose actually has that coming together in a beta form, where this oxygen is kind of on the going to the north side or up. As you see this arrow, this line here, this bond is going up, and it's on the same side as that CH2OH. We call that a beta linkage. So you can have 1,4 that's alpha, that would give you amylose, and you can have 1,4 beta glycosidic linkage, that would give you cellulose. Very, very different properties as far as polysaccharides. We can eat this, and plants use our cellulose. So just kind of neat thing to know about the differences within saccharides, polysaccharides. All right, last is our amino acids to proteins using a peptide bond. We had said it really comes together by having a amine, and it's usually in the zwitterionic form. Amines meaning amine in the zwitterionic form means that the hydrogen from here has already been transferred over to this nitrogen. So you get a negative charge and you get a positive charge typically with your amino acids if it's at neutral pH. It's in the zwitterionic form, meaning it's got two charges. So we've shown that here, but in the end, if we're going to make that peptide linkage, we need this nitrogen to lose 2 hydrogens, and it does. We go from having 3 to now having just 1. This oxygen is going to come off with those 2 hydrogens to make water, and we get a peptide linkage. This is also called an amide linkage. So that's how we make it. We make these two come together from the carboxylate end to the ammonium ion end. They come together to make a peptide linkage. And the reason why this is so important, again, we said this is going to be a step growth, but it's a very, very particular type of growth where we have very defined amino acids coming together in sequence, E, and I don't have to, and then H, and all the different types of specificity that the RNA, DNA hands to the protein to give us very specific amino acids that are put on one at a time. You can kind of see that on the next slide. So it turns out if we have a developing chain, just think of this as all the amino acids that have already been added on in a very particular way to make a peptide chain. Sorry. So this is our developing chain. At this end, we have the transfer RNA bringing in the next particular amino acid, and we can have that linkage. Now, I'm showing the mechanism, and for sake of time, there's a lot of different ways you can propose this polypeptide, or this peptide bond be formed. But in general, this nitrogen that is partially negative has to attack the carbon here that is partially positive to give us the starting point of what's going to be our amide linkage. And just please know that at some point this oxygen has to bring back its electrons down to a pi bond and kick off this oxygen here so it can reform the carbonyl, kick off that oxygen, and maybe on the way out take that proton with it, that extra one, so we have just one left. There are some other ways that can happen, and that's the fun of course doing bioorganic chemistry, is to see what's actually going on within those pockets or within the body for the transfer of the small amino acid. So we can say this is the amino acid site, just bringing one of them in, to the peptide chain that's growing. How does that actually happen? And we know that as you go on and look at the structure of proteins, it's not just about the way the amino acid came together. It starts to fold on itself to give secondary structure. We can have the whole thing together collapse into a tertiary structure, and then if you have more than one protein coming together, one here and then one over here, they can work together to make your active sites within the body for use in catalysis or whatever. All right, that's it. There's a lot of other things we could have talked about. I guess the crustaceans and their chitin shells made using some sort of derivative of your polysaccharides. Did not talk about deoxyribonucleic acids and the polymers that are formed there and the double helical structures, but just some great stuff as far as macromolecules within the body. Thanks. Bye-bye.